Very good evening to all of you. I welcome you all for today's webinar, which is a webinar on what do the experts say, a Wednesday webinar. And as you all know, from the last week, we are doing webinars on genomics. And today's topic is model systems for understanding genetic disease. And this would be delivered by Dr. Sridhar Shivasubbu. He's a chief scientist, CSIR, Institute of Genomics and Integrative Biology, Delhi. Sir has got more than one publications, and his key area of interest include genomics and precision medicine. And as has been the tradition, after the talk by the expert, we have two eminent personalities who will be discussing this topic. First is Dr. Vinod Skaria. Sir needs no introduction. He has been with us for last almost 3 months now, delivering important lectures and discussing many important topics of genetic disorders. He is a senior principal scientist, again attached with IJIB, New Delhi. We have Dr. Nikhil Sudarshan, who is a consultant pediatrician with expertise in neurology from King's College Hospital, London, Dubai. He is MBBS and has done FRCP with CCT from UK. He was formerly a pediatric lead for genomics, Southeast Genomic Medicine from NHS UK, and has played an instrumental role in embedding whole genome sequencing in mainstream pediatric clinical practice in Southeast England since 2021. So with this what's the introduction, I will hand over the stage to Dr. Sridhar Shivasubbu to deliver his lecture. Over to you, sir.
Yeah. Thank you, Dr. Mahesh, and thanks to the organizers, Dr. Shefali Gulati and the team for inviting me to the talk today on a session of clinical neurology webinar series. Please allow me to share my slides in a minute.
Asmita?
Yeah, I'm doing that.
Yeah,
I'm doing that. Yeah, is my slides visible?
Yes, sir. It's clear. Sir. Thank you.
Okay. So what I'm going to do as part of my talk is to take you through how do you use model systems for understanding genetic disease, especially in an era of genomic medicine, where increasingly a large number of people turn up with exomes and genomes in clinics, and a large significant proportion of those results are going to be variants of unknown significance. What does a clinician do, and what are the tools available to a clinician scientist, and how can basic biology aid in that decision-making process? That's the outline of the talk that I'm going to give. But what my lab really works on is captured in this nice painting drawn by my spouse, who calls it personal genomes to personal fish. So we work on humans, we work on a lot of mitochondrial diseases, we work on a very interesting subset of human genome called long non-coding RNAs, and of course we always use our favorite model organism, which is zebrafish, for modeling diseases. So these are the topics that by and large I'm going to cover. And so the areas of interest of my lab are largely looking at genetics and developmental biology, and how do non-protein coding RNA, which is a large subset of the human genome, and how does it regulate vascular and cardiovascular system. But we have sort of ventured also into a lot of neurological diseases, which I will touch upon, where we use zebrafish as a model system. But also a large part of my lab also works on trying to understand how can we use these models for personalized and precision medicine in humans. So this sort of cartoon here is pretty much a representative of what is happening in the field of genomics right now. If you do human exome sequencing, which is the most popular genomic test as of date for people who have genetic diseases, you get about 35% of results which have some meaningful evidence that the clinician can work upon. Of that 40%, maybe about 10% to 20% would have a mechanism or a therapeutic or an intervention possible by the treating clinician. But a large proportion, which anywhere varies between discipline to discipline in the medical field, anywhere between 60 to 60% to 70%, largely has what we call as variants of unknown significance. In colloquial terms, nobody has any clue what that mutation is or what it does, and therefore we bin it and leave it for eternity or future for somebody to work it up. So that brings up a burning question: should we be looking beyond genomes, that is beyond exomes? Of course, we had one of our both of our lead experts today after the talk have worked on whole genomes extensively, so they could add value to my discussion. That's one way to look at. But most importantly, how could you use functional studies for understanding what these even whole genomes tell us? That's what is the future. So let me start out with a example. Most of you are clinicians out here, so therefore let me start with giving you a clinical case, and then I'll come back to how do you model this disease. So this is an unconventional mitochondrial disorder causing infant mortality that was first came to the clinic of Dr. Madhulika Kabra and Neeraja Gupta at All India Institute of Medical Science at New Delhi, then worked upon greatly by Dr. Shefali Gulati and also Dr. Sujata Mohanty. And at the bottom of the screen are the three pillars who contributed to this: Ankit Sabarwal, Shamshuddin, both of whom have completed the PhD in my lab and moved on, and Vinod, who is a very fantastic collaborator on this project. So this was a 9-month-old baby with a history of elevated lactate and pyruvate levels, and the provisional diagnosis was Lees disease and neurometabolic disorder. The high mitochondrial score suggested that it had a mitochondrial association, but interestingly, the parents were apparently normal, and though the child did have microcephaly, small eyes, and lethargy, so clearly indicated a neurometabolic disorder with mitochondrial origins, and that's pretty much the clinical data that we had. Some more clinical data for the clinicians here. The index case here showed growth retardation, severe developmental delay, failure to thrive, elevated lactate levels in the CSF, progressive microcephaly, and many other abnormalities, finally leading to sepsis due to infection was the cause of the death. But clearly, this was a one-off case that was brought to our attention. Really, in such cases, what as a genomic scientist we do is we do mitochondrial genome sequencing. Mitochondrial genome sequencing is a very small genome. In many of our most of our cells carry a mitochondria, so we could isolate blood, isolate the mitochondrial genome, sequence it at very high coverage, and be able to use automated pipelines. In this particular case, Vinod's lab and my lab had developed this pipeline called Mitomatic, which was available for many years in the market in the name of MitoSure, sold by Eurofins. So when we ran this pipeline, we really did not find any variants that would be associated with the phenotype in this particular mitochondrial sampling that we did. And the scientific basis of doing all this was also published by Shamshuddin way back in 2015 in Human Mutation. So the short answer, we did not find anything. So we took the next easiest thing to do is to do whole exome sequencing. Again, shown on your left-hand side is the summary of the whole exome sequencing analysis pipeline. Though we did find about 136,000 total variants, when we ran down the pipeline using various established bioinformatics tools in our lab, we really did not find any pathogenic variants. The next thing in the cascade was to do whole genome sequencing. Very nice run we did, and after following the pipeline, we actually had one suspected variant that we thought might cause the or was found in the genome, and that particular variant was actually a large deletion of about 142 kilobase, covering the in chromosome 5, covering genes ERCC8 and NDUF AF2 gene. Now, I'll come back to both of these genes in a minute time, But one of the first things that when you see things like this is to go back and ask simple questions as if the gene is missing in a chromosome, the easiest way to measure is that, is this loci actually contributing? So look at the RNA expression by simple muscle biopsy. And in this case, the muscle biopsy was done, the total RNA extracted, and there was no expression of ERCC8 and NDUF-AF2. And this could be done using an RT-PCR experiment. And that was the first evidence. So I'll build up evidence towards the final conclusion. So the first evidence was that there was a deletion. The second evidence was that when we looked at the genes in the deletion and did RNA expression analysis, in this case, we used RNA sequencing, or you could use RT-PCR also if you had very specific primers. We did not find any expression for these two genes. So two evidences that I've shown you. In the meantime, the story, the way evolved was both the parents also got curious and interested in this whole affair of sequencing. The family was very cooperative. So we sequenced the father and sequenced the mother. And what we found was both the father and the mother, though they are not related to each other, actually carried the father carried a 249 kb deletion in chromosome 5, and the mother carried a overlapping yet a slightly of a different genomic interval, a 240 kb deletion, and there was a overlap between the father and mother. So the proband that we had actually by this time sequenced actually had this common area that was deleted, which covered both these genes. And if you looked at the chromosomal interval, below are shown the 3 genes that are overlapping this particular interval, and you would see both ERCC8 and NDUF actually fall right in the interval where the deletion had happened. And this is a very curious case because these kinds of deletions are not supposed to be, these are very extremely rare deletions, and you find them in 2 unrelated individuals who happen to marry each other. So how often does this happen? So that was the curious case here. In this case, the father had a additional deletion of a ULV7 gene, ERCC8 and NDUAF-AF2 gene. The mother had an NDUF-AF2 and a ERCC8 deletion. So we waited, and 9 months later, the parents again came back with a pregnancy, and we again found the same deletion in that particular CVS sample. Now, the question that we asked ourselves is, what could we have done between these about intervening 1 year period? And that's the story I'm going to tell you. Okay, so we knew that there was a deletion. A clinician had to take a decision at this point. What do you do to this infant? So, so before I go ahead with how the story evolved, here is the brief overview about the 2 genes involved in this story. NDUF-AF2 is a complex assembly 2-factor gene, and it's a sort of a molecular chaperone on chromosome 5. It's involved in mitochondrial complex 1 assembly. So it's a molecular chaperones involved in mitochondrial complex 1 assembly is what you need to remember. The gene 2, ERCC8, was a mystery for us because it's an excision repair cross complementation group 8 gene. That means it's involved in DNA repair. Just remember that. That means we have 1 gene that is involved in a mitochondrial complex 1 chaperone activity, and a second gene which is involved in a DNA repair gene. At the onset, it did not make any sense to us how these genes could be interplaying, because these genes are not known to interplay with each other in any metabolic pathway that we had wanted to study. So another side story to this, both these genes are present in the nuclear genome. They are not in the mitochondrial genome. So in a typical cell, you have a nucleus within which the nuclear genome resides. And then you have the cytoplasm in which the mitochondria resides. The mitochondria has its own small mitochondrial genome, and the nucleus has its own large nuclear genome. Okay. Now, both the genes are located in the mitochondria, in the nuclear genome. But what is known is that a large number of protein precursors originate from the nucleus, and they move towards the mitochondria, and through various well-studied mechanisms are involved in the electron transport chain, assembly and biogenesis, mitochondrial dynamics, maintaining the genome of the mitochondria, ion metabolite homeostasis, and many other activities. So basically, there is a interdependency of both these organelles in a cell. Now, when something goes wrong in the nuclear genome, okay, one of these activities, either the bioenergetics or the protein import machinery or the genome integrity or the metabolite deficiency starts to show up. And that can manifest in a high level of reactive oxygen species in the mitochondrial genome, which can be measured. So again, this is something to remember. I'll come back to it again later. So with this information in hand, we approached Dr. Sujata Mohanty at AIIMS New Delhi, who does a lot of stem cell biology. And we were able to provide her from the lab of clinics of Dr. Madhulika Kabra and Neerja Gupta and Shefali Gulati, fibroblast cells, which were cultured, and so that you can now not go back to the patient, basically use these fibroblast cells as a model system. So this is the fourth evidence I'm showing you, that how you could use evidence derived from the patient material and measure gene activity. In this case, we are measuring NDF-AF2 and ERCC8, and this is measuring the protein levels using a Western experiment, Western blot experiment. Here are the controls. The control seems to be normal, whereas you really don't find NDF-AF2 or ERCC8 to be present. They're completely abolished in the patient fibroblast cells. Remember, I showed you the RNA is already absent. I'm also showing you the protein is also absent, and there is a large deletion in the index case, and the parents are carriers of overlapping deletions. Now, since we have all the evidence pointing to a mitochondrial gene involvement, one of the things that mitochondrial biologists like to do is to measure oxygen consumption, because mitochondria is sort of the powerhouse of the cell and makes ATP. So what you could do is you could now measure basal respiration, and by adding various chemical inhibitors or activators, you could modulate this mitochondria. Okay, for example, you could shut down the ATP production, you could increase the respiration, you could do so and such fancy tricks to see if the mitochondria was responding properly. And these are all well-studied pathways. So you had the human immortalized human fibroblasts, which are the controls, and the patient fibroblasts as the experimental one, and the various inhibitors that are listed at the bottom in various concentrations were used. And shown on your left-hand side of the screen actually is the oxygen consumption on the y-axis and the time on the x-axis. And in black bars are the control fibroblasts, and the patient-derived fibroblasts are the red bars. And you would see, as you keep adding these inhibitors, the mitochondria, the control mitochondria really responds very well, as I showed you it should. Okay, whereas the patient mitochondria shown in red really shows almost a flat line. Okay, saying that they really don't respond. So we have the fourth or the fifth evidence now to say that the mitochondria actually is not functional. Okay, till now we are only looking at the molecular evidence. Here is the first functional evidence to show that the mitochondria is actually not even responding. Okay, the sixth evidence that I'm going to show you is a ultra micrograph of a TEM image of the liver biopsy of the index case. Okay, which showed altered myocyte mitochondrial morphology, which indicates hepatic stress. Because the normal, most of us seen a textbook example of mitochondria, which is nice, globular, with all the filaments and everything pointing inside. And suddenly you see these flat structures all over the place, not really looking a healthy looking mitochondria. So we have shown that the mitochondria by morphology is distressed. We have shown by a functional evidence that the mitochondria is not functional in the patients. So we have added these two evidence. Okay, that is where we started asking, can we model this in an organism? Because we are only looking at cells, and our favorite organism here is a zebrafish, because if you look at a human embryo and an early fish embryo and an early human embryo, okay, even though they are divergent by 400 million of years, okay, they both have the same genetic blueprint. And that's basically the one Bayer's law that we have all studied early on in the medical school or in the biology classes. Okay, both of them have the set of same 25,000 protein coding genes, a very well set of conserved non-coding RNAs. The vertebrate specific processes are practically conserved. You can find all organs in humans in that are there in fish also. But most interestingly, the genome size is half of humans. I'll come back to the genome size later on. So So while the reference genome was published by Sanger Institute in 2013, Vinod and I had also contributed to the wild zebrafish genome at the same time when the reference genome was published from our labs, from a very brilliant student called Ashok Patwari, who did the genome sequencing for the wild zebrafish. So we do a very simple trick. I will come back to this tricks later on in my talk, but just for now, just understand that you could take zebrafish embryos which can be produced in thousands in every day, take a small oligonucleotide, modified oligonucleotide against a gene of interest and inject it at 1 cell stage. At 1 cell stage, because these are external fertilizing embryos. And this is a control one. You can see by 2 days, that's the eye, that's the tail, and the brain and the yolk sac of the fish. But when you put in this, block the NDF AF2 protein production, okay, you start seeing deformed embryos, and at slightly higher doses, you see this gross morphology of these embryos. They don't look healthy in any way. So knockdown, this is not knockout, remember, this is just a knockdown. That means we are suppressing the production of NDF-AF2 for a brief period of time. Okay, and it can be about anywhere between 24 hours to 72 hours that the production can be blocked during development. Since the development happens so rapidly in the absence of the genes, the phenotype becomes visible. So knockdown of this NDF-AF2 induces developmental abnormalities and models severity of the phenotypes observed in proband. Okay, and of course we had looked at the protein levels. As we increase the morpholino concentration, the morpholino, you can see the signals really depreciating very rapidly. And here is a quantification on the right side, clearly saying that upon depletion of NDF-AF2, we lead to gross developmental abnormalities in an organism level. So this is sort of the 7th evidence. Now, cutting a lot of about 2.5 years of work by Ankit Sabharwal, what he showed in a very elegant set of experiment is that how this 142 kb deletion spanning ERCC8 and NDF2 genes causes mitochondrial dysfunction. So essentially what happens is that, remember, NDF-AF2 is completely abolished because it's deleted. When NDF-AF2 is abolished, it's a chaperone in the electron transport system complex 1. Okay, so the complex 1 is sort of deficient and does not function properly, due to which there is a buildup of ROS, reactive oxygen species, in the mitochondria. And whenever there is a reactive oxygen species buildup, it leads to nicking or damaging the mitochondrial DNA. But when this large amount of reactive oxygen species build up, it also starts damaging the cell and the DNA in the nucleus. Okay, that's when we realize that the second gene that's involved is actually a DNA repair gene, which is ERCC8. Okay, ERCC8 along with a complex is responsible to maintain DNA integrity. Now, when the ROS starts damaging the DNA, you don't have the DNA repair mechanism actually correcting it. So therefore, you have a double hit happening here, where NDF-AF2 actually starts the cascade of ROS buildup, and the damage built by the ROS is not being repaired by ERCC8 because that's also abolished in the patient samples. Okay, so really it made sense why these two genes were, when damaged, when deleted, was leading to an infant mortality and not a very early lethality of the embryo. Because again, remember, there were some levels, because it's a chaperone, NDF-AF2 is a chaperone, so it could somehow sort of override. There are other chaperones in the system that can sort of help keep the ATP generation machinery going. But as the embryo grows, you start feeling the pinch of the abolishment of NDF-AF2 and ERCC8. So in a nutshell, what I've showed you in the 9 months that intervened between the two babies that came to All India Institute of Medical Science was a series of experiments that took place between IGIB and All India Institute of Medical Science, where you had the patient fibroblast. We could do a mitochondrial functional assay to show that the mitochondria was completely non-functional. You could do a zebrafish experiment to really show the organism level difference. But most important, what happened is that we also built an MLPA-based diagnostic assay. Again, I'm not going to show you data for that. There's a population level screening that was done, then the extended family screened to figure out where is this deletion originating, because you cannot do genome sequencing for every individual. An MLPA assay is a pretty robust as well as cheap and affordable assay that can be now rolled out to family and community level screening. So we could build from a clinical diagnosis, from a provisional diagnosis to a actual genetic diagnosis to actually doing a prenatal, getting the functional studies up, building the animal models, building the molecular genetic understanding of it, creating a cheap and affordable assay for population screening, and finally leading to a full-end understanding of the disease process. But that's all fine. But could we do more beyond genome and exomes when we have more such patients showing up in your clinics? That's the whole gist of the story. To do that, here is my next set of part B of my talk, where I'm going to talk about the zebrafish. Okay, the zebrafish is essentially the key here, because you could do a lot of things that really you cannot do in human beings readily. This could be used as a sandbox. And zebrafish is an Indian import to the West. It's found widely in India, but not extensively used in India till recently. Till about 10 years ago, it was not extensively used. I would say 10, 15 years ago, it was not extensively used, but extensively used abroad. So that, so people like me, when we realized this early on in our PhDs, discovered a way to go do our postdocs in labs abroad, learn this part of zebrafish genomics and genetics, and bring it back to the country. And today we have a number of labs, about 12 labs in the country that can do this work fantastically. So a typical zebrafish lab that you see in your screen here can house about anywhere between 50,000 to 80,000 animals, and that's a large number of animals. This is one of the largest facilities in the country, run at IGIB. There are at least 3 such facilities in our country. You can generate a large number of transgenic animals. I'll show you some examples of it. Again, coming back, why do we use it? Because I've already explained, okay, there's a very conserved set of processes between human and zebrafish that allow us to study, go back and forth between these 2 organisms. And one of the reasons why we are able to use zebrafish is largely because of the optical clarity that we see in zebrafish, and the clarity is very visible. This is a 2-cell zebrafish, it's the external fertilizing embryo. So if you can put it on a dissection microscope, you can actually observe this by thousands every day. So you can, so the exciting phase of human biology actually happens in the mother's womb, whereas here you're able to see that exciting phase in reality under a microscope, in a dissection microscope. And this is the first 16 hours of zebrafish, from going from one cell to the head at the top of your screen and the somites and the tail at the bottom of your screen. You can see that's how fast the zebrafish develops. We can we generate anywhere between a few hundreds to 1,000 embryos by setting up a few crosses, a few breeding crosses. The embryogenesis is completed in 48 hours. And the major organ systems are developed in 48 hours, as you can see in the bottom panel here. So basically, experiments are about 48 hours to 72 hours long, and in a few thousands of numbers, and a lot of fantastic tools available for manipulating their genomes. Like I said, Vinod and Ashok were key to getting the wild zebrafish genome sequenced way back in 2010 that we published later on, essentially showing that zebrafish are not clonal populations. The wild zebrafish are not clonal populations. They are as much divergent as humans are, and the human disease gene orthologs are very well conserved and do not display enrichment for variations. So now, how do you manipulate when you get genomes, when you get zebrafish? It might sound very trivial, but believe me, it is as trivial as this. For you can take a 1-cell zebrafish embryo. You can use a number of tools that are now available extensively and very popular. You can just inject simple GFP constructs to zinc finger nuclease, to knockout genes, to talons. I'll show you examples. Or CRISPR-Cas9, again, I'll show you examples of how those can be simply injected and into 1-cell zebrafish. And that's a very microscopic needle being stuck into a zebrafish embryo. You can see the 1 cell, the yolk, and the chorion around it. And in about 24 to 48 hours, you'll see the resultant. In this case, you have injected a GFP, a construct for making GFP protein. So you see a green fish, or you inject certain particular constructs that express only in the brain. You can see the brain nicely labeled up in red fluorescent protein. So you could do this fancy experiments and generate a lot of colorful animals that can be studied on your favorite animal. And you could also do very unbiased screens. And this is something that I have done as part of my studies, postdoctoral studies, where you could make this fancy yet complicated constructs that could do conditional knockouts. And this image that you are seeing on your screen represents the first conditional mutant alleles outside the mouse model. Mouse is known to generate a lot of conditional mutants. That's what it's famous for. And zebrafish is sort of the second conditional models that were generated and extensively used by labs around the world. So because you are all neurologists, I decided to sort of pick this example. And Vinod and I had worked on generating a lot of conditional mutants across the neuronal tissue. And as you can see in the right side is a sort of a diagram of a zebrafish brain, and you would recognize the telencephalons and all the subparts of it. And you can see you can generate very tissue-specific expression using these conditional alleles, whereas in particular parts of the brain, and be able to study. For example, this particular line that we are interested in is a protein trap line called GPT1365, which labels up a single neuron that originates somewhere in the between the midbrain and then radiates out from there. So you could be as specific, and all this, remember, is done under a simple compound microscope, fluorescent compound microscope. Doesn't require very fancy tools to do that. You could also, I showed you an example of how you can inactivate genes temporarily using something called the Morpholino or a modified oligonucleotides early on when we blocked the NDFAF2 gene. So these are small oligonucleotides, about 20 base pairs to 25 base pairs long, that can target the RNA or the DNA, depending upon how you design it. Basically designed upstream of the translation start site, and prevent the initiation complex from reading through it, basically physically blocking the initiation complex from reading the protein RNA sequence into the protein. Therefore, the protein is not formed. So for example, this is a wild type zebrafish, 2-day-old. You can see these black patches all over the place. There's nothing but pigments. The reason why I selected this is so easy to understand. You could generate morpholinos against a gene called vacuolar ATPase, and you can see the pigment completely getting decreased. And if you had something, a morpholino against tyrosinase, which is a gene involved in pigmentation, you can almost create an albino zebrafish. So it is that easy, but these are temporary experiments, to quick and dirty experiments to get the hypothesis validated. You normally have to follow these experiments by doing extensive knockout studies. But before I do that, the other area of interest, like I told you, is to understand long non-coding RNAs in humans and zebrafish. The reason why we are interested in this particular subset, and the reason why I want to introduce you to this subset, is because almost 50% of the human transcriptome is this red color bar, which is nothing but a subset of RNAs called as long non-coding RNAs. So the RNA that makes the protein are called protein coding RNAs. They are about 1 to 2% of the genome, and a large subset of them would become long non-coding RNAs, which don't make protein but remain as RNAs and modulate biological function. So, and they are spread across evolution and found across all animal systems. So again, when we were studying, Vinod and I were studying the long non-coding RNA subsets that map to the zebrafish brain. For example, at 24 hours post fertilization, if you looked at a zebrafish brain, those are the two eyes. That's the forebrain, midbrain, and the hindbrain. This particular LNCRNA called BR LNCRNA1 actually the expression is found throughout the brain, but by adult stage, that expression is restricted to only the cerebral crest, as you can see shown by the pointed arrows. So you can see these long non-coding RNAs are very, very dynamic, tissue-specific, and control gene expression. So they become very important subsets to study. So Paras and Samatha, who are PhD students in my lab, actually took up the set up the whole process of wanting to study this. And in this case, we used a gene editing tool called Talens. CRISPRs have gained a lot of popularity because of their ease of use and the recent Nobel Prizes and the controversies around it. But preceding that, there are very fantastic tools called as Talens and zinc finger nuclease that are extensively used for over 20 years. We have been using them. And in this particular case, Talens were targeted against a particular long non-coding RNA to create small insertion deletions shown at the bottom of your screen here. So these insertion deletions, one of them were selected in a zebrafish, and I'm drawing your attention to the bottom of the screen where you have the three green zebrafish, fluorescent zebrafish. So in green color is the vascular tissue, the endothelium labeled in green fluorescent protein, and in red color is the red fluorescent protein labeling the red blood cells. So we have this fish to understand vascular biology in our lab, and you can look at the green channel, the red channel, and also do biochemical staining for hemoglobin. And you'd notice that in the control animals, the brain is largely devoid of any flow of blood in an irregular manner because of the very strict blood-brain barrier. Whereas when you knock out this LNC RNA using Talens, you suddenly see a large bleed in the brain or the hemorrhage in the brain. So one of the golden experiments that geneticists do is actually put back the RNA externally, and it should revert the phenotype. That means it should basically correct the phenotype. In this case, the phenotype is a hemorrhage, and you can see a reversal. So that's sort of quick and dirty experiments that we do to understand if the particular gene that we deleted actually caused the phenotype. The long story short, Paras actually ended up finding the human counterpart, and the human counterpart does not have a sequence similarity to the zebrafish counterpart, and that is a characteristic signature of a long non-coding RNA. But he was able to do extensive functional work to finally really go to the molecular evidence to figure out how does this LNC RNA called as vascular endothelial associated LNC RNA, or called VEAL2, interacts with a very well-known studied protein called PRKCB, protein kinase C beta, which is involved in endothelial cell permeability, and how this interplay is involved in maintenance of the basal endothelial permeability. For example, what he showed is how in a normal cell, in response to VEGF, the VEGF receptor receives the signal, and through a well-defined cascade actually signals to a metabolite called diacylglycerol. Diacylglycerol actually binds to PRKCB and modulates the activation of PRKCB, or moves it to the membrane, and the PRKCB then actually phosphorylates junctional proteins to modulate the junctional integrity or the flow of metabolites and oxygen and blood to the tissues. So in hyperglycemic conditions, there is an excess of glucose, and excess of glucose actually causes excess of DAG, the diacylglycerol. And in the presence of excess of diacylglycerol, PRKCB is constantly hyperactivated, leading to hyperphosphorylation of the junctional protein, leading to a complete degradation. And VEAL2, which is our long non-coding RNA, actually regulates PRKCB and actually sequesters it and makes it inactive. So in this case, PRKCB is not able to sequester and make VEAL2 is not able to sequester PRKCB and make it inactive because there is a hyperactivation. So what this study really showed was that how a long non-coding RNA could regulate interaction between a metabolite and a protein coding gene and be able to make it inactive. So this work was published, as you can see at the bottom of the screen, as a journal cover page, and very well received because this has a number of firsts in the field, which people in the long non-coding RNA field would more appreciate because this is sequence independent functional conservation was seen in this particular case between humans and zebrafish. So summarizing what I have presented to you till now is that one could deliver precision medicine beyond genome sequencing, because genome sequencing is largely a commodity now offered by many clinics. But what happens when you don't have an answer to the sequencing that you did? Many times in genetic disorders, you could use organisms such as zebrafish or tissue culture settings that I showed you to actually create patient-specific models, use that patient-specific model to get patient-specific data, and at some time point use that patient-specific data to actually drive patient-specific therapies. So in this particular case, instead of a therapy, we actually, since we knew the mutation and we had the MLPA test, there has been subsequently 2 pregnancies in the family, and in both the cases, the MLPA tests and genome sequencing have been extensively used along with the functional evidence to actually have healthy babies. The eldest of them is in class 2 now, and the younger one is just entering UKG. So you can see how genome sequencing combined with experimental evidences can really deliver precision medicine to the families and patients. So here is a small tidbit that fish is not, it is not just a fish, it's a hope for many patients. And hopefully in the years to come, we'll all have our own personal models or personal fish that we'd like to hold on to, because that would become key to our disease therapies and diagnosis. I'll stop there and acknowledge a large number of students at IGIB whom I have sort of mentioned throughout my talk, who have contributed to this fantastic work. This and beyond this, fantastic collaborations from Vinod, clinicians from AIIMS New Delhi, and several other hospitals in and around India, researchers, patients and their families, very important contributions that they have made. But most importantly, fantastic and consistent funding from Council for Scientific and Industrial Research, really starting from a single genome in 2008 that Vinod and I sort of published and demonstrated that you could do, which was the 6th genome in the world, all the way to really putting it into patient care, was a sort of a dream come true for all of us and for many of our students. I'll stop there and take questions. Thank you. Back to you, Doctor.
Yeah, thank you, sir. Thank you for a very wonderful talk. Now we'd like to request our expert, Dr. Vinod Skaria, to give his comments on this talk and his views. Dr. Vinod.
I think Dr. Nikhil can go first.
Okay, fine. Yeah, Dr. Nikhil.
Yes, yeah, thank you so much. Thanks for this opportunity to join you all. Before I actually go into the talk, I just have an honest admission to make. This is to Dr. Sridhar and Vinod. This is to say that every time I see your presentations, my mind is blown away, literally, with looking at the amount of fantastic work you've done over the last few years. And I was telling Vinod last night that you've probably done so much work that nobody can do in a lifetime so far, to be honest. And I do hope at some point you get the highest national and international recognition that one can get. And thank you so much for this fascinating talk again, Dr. Sridhar.
Thank you very much. I appreciate your comments. Yeah.
Thank you so much again for this fascinating talk, and it was fantastic to hear about the fascinating story of mitochondrial diseases and functional validation. And what was again shining through in these examples was the high dedication and commitment of clinical and academic teams, talking back and forth and chasing the diagnosis to a meaningful point. Always fascinated by this, the utility of zebrafish genetic model. Fantastic, great thing. So when I was called for this session, and when I sat through the beginning of the session, I was having a lot of imposter syndrome in my head, thinking, what am I doing here? This seems so complicated. I'll just tell you what my background is and how this is relevant for clinicians like me. So I was closely involved in implementation of widespread use of whole genome sequencing in Southeast England. So the NHS UK has come out with this mission statement that they want to implement and make whole genome sequencing available as a mainstream clinical test. So just like you do an MRI or a blood test in future, whole genome sequencing would be one of the early tests that will be done by clinicians. So my work was specifically on persuading clinicians to think about genetic conditions to avoid the diagnostic odyssey for patients and to utilize genomic testing early on. And a key part of our work was about counseling families about variants of uncertain significance. So that's why this talk is so relevant to clinicians and implementers like me. So what was very much of an eye-opener when I looked through Vinod's presentation from last week, the fact that the hitherto published literature on phenotype to genotype correlation may not be always accurate, and we may have analyzed and interpreted wrong genetic changes as clinically significant. So that was quite an eye-opener. And together with that, when I looked at the variant annotation using literature search process, I was again worried about that process because you are presuming that what is available in literature so far is the truth. So that's bound to have add-on bias after the other. But this presentation, for that reason, gives clinicians lots of consolation that there is now an objective functional validation where you can prove using multiple ways and multiple times and replicate that the gene variation you found is truly pathogenic. And similar to the mitochondrial stories we also had cases where patient waited a long time for a diagnosis, and eventually from the 100,000 genome project, for example, we found a variant in LRPPC gene, which, as you would know, causes Lee syndrome, a French-Canadian subtype. But when we found that, there was an issue with what we made of it, because the child's phenotype did not fit in with that. For example, there was no lactic acidosis, there was no basal ganglia changes on MRI. So there was a tendency among clinicians to reject that finding. But what worries me is our pre-existing description of phenotype is made by biased interpretation of what has been reported so far. So that child still now has not got a concrete diagnosis because we don't know how to, and perhaps that's where the functional validation comes into place. So before I finally open up to the questions, I just want to highlight a few ways in which clinicians work and how we can improve this process of joint working with scientists and geneticists. So in clinical diagnosis, there's a lot of cognitive bias that goes, especially about interpreting test report. For example, like 10 years ago, when I used to see a microarray report, I had a tendency to tell patients that your child's condition cannot be due to a genetic problem because your microarray is normal. So that was a bias, thinking that that test result is the ultimate. So, and unless the clinician is curious and wants to speak again and again to the scientist and geneticist, that cognitive bias would be a hindrance to diagnosis. And similarly, when we discuss variants of uncertain significance, now we learn to modify our conversation, saying that at the moment, with the available information and genetic technologies, we are not able to say what the diagnosis, or even if we get a negative diagnosis from whole genome sequencing, we say with the available technology, I haven't found a reason for genetic reason, not saying that your child does not have a genetic disorder. So the other thing I wanted to highlight is all the fantastic projects that you've done in India. The key element that's shined through that is the clinical curiosity of the clinicians and the academic curiosity of scientists and geneticists like yourself. That curiosity and proactive communication is what we need to advance this area of medicine. And finally, is also reading about the future of genomics. And one of the questions I want to throw up is the idea of revisiting variants. How often should we revisit variants? And one of the leading geneticists from the US said, just like you have an annual health checkup, in future, we might need to do an annual variant update or genomic update in future. That might be the future. So thank you so much for the fascinating talk. I'll stop talking and open up for the questions. I do have my own questions. I can open up with those questions if that's okay with you, Dr. Sridhar. So first question I wanted to ask you was, this seems amazing, but do all labs do functional studies before they produce a report, or does the clinician has to repeatedly poke and persuade, or is it quite variable according to which lab is serving you?
So under the Guardian program, the Genomics for Understanding Rare Disease India Alliance Network, it's a research-based program. So we would not take up cases that has an obvious answer available or a test available, mostly in the commercial domain. And it's also, like you mentioned in your own talk, that it's a two-way process. The door opens both ways. The curiosity should come from the clinician as well as the geneticist, and most importantly, the patient should also be cooperative. Okay, in this example that I showed, over 4 years, the parents cooperated, and so that kind of involvement across at least 4 or 5 labs, the patient is required. So in a nutshell, it is not a straightforward exercise. It usually clinicians have an expectation for quick turnaround time, which does not happen because these are research-based studies. Like I said, even in the best of research labs around the world, no more than 20% of the functional studies are have a definitive endpoint. So, clearly the rewards are very slow, but they are very rewarding in the sense that if you have a functional evidence and you convert it into a population scale screening assay, suddenly you have a few thousand to almost 1 million subpopulation of that particular ethnic group benefiting from it. So we have had examples of a mutation that Vinod's lab and my lab worked on along with Dr. Shefali Gulati, was this an Alband mutation this was MLC1 gene, which is a completely new mutation found in one family, but we have 50,000 people in the same community suddenly benefiting from it with carrier screenings. So it is a two-way process, definitely. Not all projects that we start end up in successes, but like you mentioned, we revisit them once in a while. Whenever there's a new publication, we must have hit a dead end, but then there is the small thread of positivity that shows up. We go back and quickly follow up and see, did we miss out something there? That curiosity is going to really bridge the gap, is my feeling in the long run. But yes, getting what I call as quick and dirty tests. Okay, for example, the mitochondria, getting a biopsy and a fibroblast culture and the oxygen consumption assay. Okay, if it could be established in a mitochondria or in a pediatric lab that sees a lot of mitochondrial cases, it could be an assay that could have a very defined turnaround time. Or a zebrafish assay that you found the mutation, can you quickly go back and knock it down? That's about a 1-week assay. Okay, so we need to establish the screening assays, high throughput screening assays. Not that the data should be very clean, but you need significant data so that you can proceed ahead with it. Back to you, Dr. Nikhil.
Thank you so much, and I hope this will become a mainstream practice, because we're going to see an explosion of genetic tests being done in various parts of the world now. The NHS UK making it mainstream. Every clinician is going to send them right, left, and center. So hopefully this will have to be a mainstream process very soon. Thank you. Sorry, quickly, before I hand over to Vinod, can I quickly ask, do you see the emergence of artificial intelligence and AI will have an influence on functional prediction by avoiding animal models like zebrafish? Can we recreate that using artificial intelligence?
The very fact that the artificial intelligence requires a training data set. Okay, so for example, it can be AI can be really used well in annotation exercises for, let's say, ACMG annotation of variants or predicting the pathogenicity of variants. But it'll stop there in large ways unless you have functional assays. Like in my single talk, I showed about at least 3 independent functional assays. I'm not saying that all of them have to come in, okay, but at least have a medium throughput to high throughput functional assay that can be done at a very efficient manner. I think the AI is sort of the limitations would end at the functional assay stage. But of course, if somebody were to really go back and mutate every residue there in the protein and build that evidence to what happens when the residue is mutated and use that as a base to now predict what happens to the nth residue, okay, yes, then AI becomes useful. But somebody has to do that bull work.
Yeah.
So the AI is only useful when you do that background work.
Okay, thanks very much. I'll hand over to Vinod now.
Yeah, thanks, Nikhil. And yeah, I have nothing pretty much to add. Probably I will ask just one question to Sridhar so that he can also give a perspective to clinicians. Probably 2 questions. The first question is, how do a clinician work with a scientist, and what does it take to work with a scientist? And second, given that genomics is going to explode, a lot more genomes are going to be done, how could a clinician and a scientist sit together and prioritize which variants that needs to get a functional validation and which variants should not?
Okay. Before I say that, I think there is a question in the chat box for Dr. Nikhil. So while Dr. Nikhil is reading it, I'll answer your question. So first, what does it take to make that dialogue happen between a clinician and a geneticist or a molecular biologist? I think the large component has to be to do with trust and patience. Okay. These 2 are the 2 pillars that I really say. Each of them have to trust each other. Each of them need to have a lot of patience, because what I've shown you in my simple example is almost about 3.5 years of work to solve one single rare disease problem. Okay, and there are close to about 8,000 of them out there. So that trust factor and the belief in wanting to solve the problem and not wanting to hand over a report to a patient, because handing over a report to a patient happens from a service provider. Okay, a large number of my collaborations fall through because initially clinicians probably think it's a service that we give out and they want to get the services done, but finally they realize that it is not a service, it's a research program. In a research program, there are no negative research reports going to be shoot out, because we ourselves don't know what is negative and what is positive. It's a matter of trial and error that we together work. Like you saw, 4 clinical labs and 1 bioinformatics lab and 1 molecular biology lab got together to solve this. So it takes that kind of a patience. So of course, sustained funding is something that is important for both the labs to work together. I would really, it really helps taking the as the next steps together. Now, that sort of summarizes the discussion between the working relationship between a clinician and a molecular biologist in large ways. The next question that you asked is, can you repeat the question, Vinod? What was the second question about variants? I think I might have lost Dr. Vinod there. But anyway, I think he had asked me the question that how do you qualify variance? Anyway, I'll wait for him. I think I missed part of that question. And back to you, Dr. Nikhil. I think I'm.
Yeah, the question was which variants to choose.
Yeah, okay. Which variants to choose for functional validation? Good. So there are a number of criteria that we use when we want to choose a variant. And usually, interestingly, these are not the variants that show up on the top of the list for a clinician. And that's been my learning, is that let's say you do an exome sequencing or a genome sequencing, you have a bunch of variants on the top. And usually that's what people start working. No, we don't do that. We actually go back, really understand the clinical phenotype. We actually quiz our clinicians or almost torture them to the extent to give us every shred of evidence that ever has been built on that patient. And sometimes clinicians do get bugged by our constant poking them. Sometimes because embedded in those random tests would be that one single evidence that would point to a gene misbehaving, okay, which would have been a standard test did for some other reason. Okay, or it would be a panel of biochemical tests that the clinicians ran. They saw something that was off, but there would be one single evidence buried in there. So we really like to request clinicians to give us even something that is non-relevant, insignificant. Please do submit, don't leave them out. And because that is where you build evidence. Any small observation that you have about the patient, please note it down, pass it on. We would like a very detailed deep dive into the clinical description of the patient and use that to actually drive the biology that we build the hypothesis on. Okay. And like you said, I had two independent genes, ERCC8 and NDFAF2. One was a mitochondrial chaperone and the other was a DNA repair gene, both completely not related to each other. But very early on, we knew that they would be working together. And there were a number of other experiments that I did not show, actually, that we did to prove that they both were involved. And kudos to the clinicians who had provided that evidence very early on to us, saying that there might be a DNA repair that might be also involved. So it takes a lot of effort. Now, how do you choose variants from this few thousands of them that show up? First is believe where the data takes you. Don't pre-build your hypothesis. I tell all my clinical colleagues, just because you have your favorite hypothesis, don't go after them. Go where the data leads you. That means go where your bioinformatics leads you. Make sure a bioinformatics pipeline is super tight. Make sure you're using the proper ethnic controls. For example, if it's an Indian patient, don't go and look at the Caucasian population manuscript and say that, oh, I think now my phenotype matches, therefore I found my gene. No, that's not what you do. You actually go back to your ethnic controls, which are either the Southeast Asian genomes that are part of the larger 1,000 genome programs, which is very less, a couple of hundreds, or you look at larger programs such as the INDIGene that we know, then I have 1,029 genome sequenced and allele frequencies are publicly available. The genome HTS sequence, because again, if you're in this part of the world, there are 512 Indian samples there. So talk to the right people, get the ethnic controls, compare to the right control. Don't compare apples and oranges, compare apples and apples. That's the analogy. Third is, once you have had your watertight bioinformatics, you should not have more than about 2 or 3 variants available for you to work on. Once the variants are available, build the hypothesis. Don't shoot in the dark. Okay, build relatively easy screening assays, or what I call as modest throughput screening assays. For example, here the modest throughput screening assays were actually the zebrafish ones. Okay, though in the sequence of events I did show them later on, but those are the easiest one to build because all I had to order was a 20 base pair oligo, modified oligo. Okay, it cost me about 22,000 rupees to 28,000 rupees to order one. Okay, it is expensive, but given a stem cell culture fibroblast experiment, this is way cheaper than that. That was going to cost you something like 2 to 3 lakh rupees. Okay, and upwards from there on. So, and you get the answer in about a week's time that does this gene actually cause such gross phenotypes. Okay, so build your modest throughput screening assays. And for your favorite, so I also work on a disease called as cardiac channelopathies. Okay, many of you, they're ion channel deficiencies. Very easy to measure electrical currents. So use such assays that are very easy to measure. We also work on other diseases where we use RNA, DNA, proteins, anything as a surrogate assay to measure. So build such assays and try to work with the where the evidence leads you. Most of my clinical colleagues, unfortunately, would read a paper, come back to me and say that this is what is thinking is happening in the patient. And usually that is not what is happening in the patient. Has been, I had to reprove it wrong. So that has been my challenge working. But once you convert them, they lifelong remain converts. So that's what I would do. But also if you see a very peculiar looking mutation, like a deletion in a region where it should not be, okay, use your best clinical or the scientist hat to justify why do you think it's going there. The data is telling you something. Lead where the data goes would be my advice to the clinician, and let the variant emerge out. Don't pre-decide the variant. Yeah. Back to you, Vinod and Dr. Nikhil.
Thank you. I'll address one of the questions on the Q&A. It's been addressed to me, but I'm not sure if I'm the right person, but I'll just address it anyway. So it's a question from Keshavan Markandan. Keshavan has asked when should researchers in India should focus on gene therapy for rare diseases. So thank you for asking that question. Probably Dr. Sridhar and Vinod will also comment on this, but my feeling is just like the explosion in the uptake of genomic tests we are seeing and functional validation and things, in parallel we are also going to see processes like gene editing for gene therapy in rare diseases, as Dr. Sridhar has also shown just now how gene editing can be extrapolated to therapeutics. So my humble opinion is we are going to see an explosion in gene therapy for rare diseases as well worldwide, and I have no doubt that India will be pioneering that as well, seeing all the fantastic work you've done. But I'll just ask Dr. Sridhar and Vinod also to comment on it.
Thanks, Dr. Nikhil. So yeah, I do echo your thoughts that it's a very promising era where you could do n=1 trials, and the tools are, I think, right about where they need to be, especially with the CRISPR-Cas revolution happening. This is not the first time it is happening. There has been a series of gene therapy tools about for 20 years that has been in making. So this was sort of culmination of a lot of stuff. So I do see the n=1 trials leading to a lot of gene therapy applications in rare diseases. In India, like my colleague at IGIB, Dr. Debojyoti, is building CRISPR-Cas9 based tools called the FN-Cas9 for sickle cell anemia and a couple of other diseases. My other colleague called Shiva Prakash Ramalingam has been building for sickle cell and thalassemia. He has been building gene therapy tools. So I'm sure there are places in CCMB at INSTEM and other places in India where we do, and worldwide also we are seeing a explosion of publications. So it's just a matter of time where you would start seeing them in real life also. Back to you. I think you're mutated, Dr. Nikhil.
Yeah, so if I may bring up another topic, please. It's about the variant interpretation during newborn screening program. So, you know, there are several places where newborn screening trials are being launched. And one particular challenge, I suppose, in those things will be there wouldn't be a phenotype to go by because we haven't seen the phenotype yet. So, and I suppose the functional validation studies will become even more important in such scenarios. So, any thoughts on how the variants from newborn screening projects where will it lead you?
So, so as far as newborn screening goes, screening can practically be done only when you have evidence already in place. So unless there are these rare cases like the ones that I have presented or others are doing, a large public health application of newborn screening would largely happen on variants that have been well qualified and have been known to cause the disease and are prevalent in the community or the country where they are being implemented. So I don't see a large scale requirement for functional evidence building for newborn screening assay. However, if the screening assay throws up certain variants that don't have evidence, they become templates for building the next generation of new biology. So one should be able to differentiate between these two, or the reporting has to be very clear that the reporting happens only for well-established variants, while the opportunity to look at this new emerging evidence or new variants that don't have evidence is pushed into the R&D pipeline readily. They are not ignored. So both have to go parallelly. I think Jayant in the audience has raised his hand.
Yeah. Hi, sir. First of all, it's fantastic to listen to your talk. My question is more of a research case. What I wanted to know was that we talked about lack of genotype to phenotype correlation. So when we talk about, we have started working on cleft lip and palate, and we see very rare examples where you have the same mutation that leads to a different phenotype. Not different, there's slight variations in the phenotype. So do we take in, so in that case, we generally take into account epigenetics as a component, right? So what are your views about how important epigenetics is? Because what we have been, studying is that whether, what you talked about was the genetic component. Can we talk about bridge a gap between the genotype and the phenotype, talking about the epigenetics of rare disease? And are we venturing into that? Are we doing any kind of work with respect to epigenetics to connect or to bridge a gap between the lack of correlation between phenotype and genotype?
So while your thought process of wanting to use epigenetics as a missing link is very valid and an area where the research labs are venturing in, but I think their application in the clinical space is I don't see it readily happening, primarily because the evidence has to be built in place. Right now, the evidence for genetic variants are very strong. Okay. And they have been studied for a few decades at an end now. So I think the epigenetics is an interesting and a very, very important subcomponent of the heritability that we need to build the evidence before we push it into clinics. I don't think the evidence except for a few handful of stuff, is there in very strong evidence exists for us to put it into clinical practice. But of course, there are very handful of stuff where the evidence has been generated. So those are valid. Otherwise, I think researchers like you should develop the evidence and then sort of take it into clinics.
Thank you. Exactly my point, sir, because I was thinking that when understanding the genetics of a disease can actually help you solve the question, where does the epigenetic component comes into play? So you've answered it right. That yes, I mean, of course, that we need to develop more evidence for that. Thank you. Thank you so much.
Yeah, thanks. And there's a question in the chat box. Dr. Nikhil and Vinod, can I read it out? Dr. Mahesh?
Yes, sir. The question by Dr. Akshata, sir. Either precision therapy for mitochondrial disorders in India. Precision therapy for mitochondrial disorders.
So the short answer is precision therapy is not yet, not that I am aware of for mitochondrial disorders. We are still, for mitochondrial disorders, in India, we are in the sort of screening phase, trying to figure out very effective and affordable screening tools for mitochondrial disorders, because there are 25 million babies who are born every year, and there is a defined proportion of those babies who do show up as mitochondrial disorders in clinics. About 5,000, 1 in 5,000 babies do have some mitochondrial disorder that show up in clinics. So I think right now we are more in the screening phase and some bit of research phase, like the one story that I showed you. There are some exciting stories happening in India. Therapy not there yet, but I'm aware of precision therapies being built for mitochondria by some of our collaborators in the US and the UK. So I am aware about it, but it's all the building blocks are there. We still need to build it for patient care, which probably will happen in the next few years.
Thank you so much, sir. Thank you for a very wonderful discussion. I'd like to ask both the experts for the final comments. Dr. Nikhil.
Thanks again, Dr. Sridhar and Vinod and Mahesh. I think we are truly in an exciting phase in medicine. I keep telling my colleagues and residents that very soon medical textbooks will have to be rewritten in big ways in the next 2-3 years, because all the pattern recognition and phenotypic description we've made so far is based on the ignorance regarding genetic discoveries. So all that will change, and truly excited to see what's going to come in the next few years. Thank you.
Dr. Vinod.
I think he's having some connectivity issues. I'm not. There's a question there on the chat box on what is Sridhar's doctor's take on genetic cause of cerebral palsy. I'm not the best expert for this particular disease. So best is that I don't answer, do a guesswork here. If somebody could write to me, I can do my homework and get back. I'm not aware about the genetics on cerebral palsy.
Dr. Vinod.
I think we.
I think he's got some network issues.
Network issues. He's not able to.
Yes.
Yeah.
Sir, last words from you. What's in the future for us on this?
So I do see Dr. Nikhil actually mentioned parts of that also. I do see a large scale implementation of genomics leading into what is this, some new field called genomic medicine, because there's a lot of evidence that have been generated, but not actually put into practice at the genomic scale. And if that could be done, for example, I see a lot of clinicians actively using exome sequencing. But remember, exome sequencing gives a lot of information. They practically look at the 2 lines written by the service provider and keep it away. My suggestion is not to do that. Maybe at least ask for the raw data from the service provider or your own hospital system. Hold on to it. Like Dr. Nikhil said, maybe once a year, have a mechanism to go through a bioinformatic pipeline. I know it is sort of daunting to set it up, but it's not that daunting, because I myself, between Vinod and me, have trained a few hundred clinicians. So we know that a good 15-day training is very good enough for you to do that exercise. You don't have to be an expert to run down, to set up a small database or of your own exomes of your patients and go back and check it. Also, try to sort of build the evidence over time. So I do see a lot of exciting times for genomics and people involved in genetic medicine, because it is going to explode. So while it explodes, you better learn it and be there when it happens. We are in the genome generation, so we should be able to use the power of genomics. I'll stop there, and thank you to all the organizers, Dr. Nikhil, Dr. Vinod, Dr. Mahesh, everybody else, Dr. Smita, for the fantastic interaction that we had. Thank you.
So one more before we conclude, I want to know what disorders you're working with now, as of now in your lab, on what
Pardon?
conditions you're working, the functional studies.
So what diseases my
What
lab
is it?
personally works on cardiac channelopathies and mitochondrial disorders. These are two favorite things that we work on. But
Okay.
usually we don't say no to people when they have a very good patient cohort and worked up cohort. Because within the Guardian program, we have had 280 clinicians, researchers. So even if something that we don't work on, we would practically know somebody else who has the expertise. So we are able to connect those dots and direct them to the right people. Like we have people with expertise on repeat expansion disorders. We have people who are very good at neurological diseases of a particular kind. So we are able to direct them to the right places. So that's something that we usually do. Primary immunodeficiency is another area that Vinod's lab and my lab extensively works up. So do write to us. We would be happy to discuss further courses.
Thank you. Also, one more comment by Dr. Sonalata. She wants to know about the course details.
Yeah.
When will this come?
Yeah, the course has been sort of put on hold. The physical course, the one that we had, the 15-day hands-on course, has been put on hold because of derailment by COVID, largely because we lost that part of the funding during COVID, and we have not been able to restart it, unfortunately. But the good thing that has happened is that we have, and largely the credit goes to Vinod here, not to me, who managed to put the most of it online.
Yes.
So between Vinod's lab and my lab, we run this annotation course, genome variant GVCI course, genome variant annotation and interpretation course, which is completely gone online. And we are happy to see that earlier we were catering to 7 clinicians, 10 clinicians per year, and now we are catering to 3,000 clinicians in a single go. So clearly that is the way to go. So instead of doing a hands-on course, what I would say is engage on the online platform, learn yourself, and then if required, you can always come down for a very short visit. Wherever we are, we would be happy to help you.
Thank you, sir. Thank you, sir. I'd like to thank our speaker, Dr. Sridhar Sisubo, for a very wonderful and engaging talk. I'd like to thank both our experts, Dr. Vinod Skaria and Dr. Nikhil Sudarshan, for their valuable comments and inputs. I thank all the delegates who attended today's webinar and made use of this wonderful talk by Dr. Sridhar Sisubo. We'll have another talk again next Wednesday at 8 p.m. The details of which will be put on both our email and the AOC India WhatsApp group. Please follow your WhatsApp and email. We'll be giving the details of the next week's webinar. I thank all of you again for attending today's webinar, and thanks once again, Dr. Sridhar, Dr. Vinod, and Dr. Nikhil. Good night.
Thank you, and good night to all of you.
Yes.
Thank you. Bye.
Good night.